Good afternoon, everyone. And for those of you who don't know me, my name is Sophia Bourgeois, and I work in the Advancement Department at the Burke Neurological Institute. I wanted to start this meeting by taking a time to thank you all for being with us today. We at BNI understand that this is a really difficult time for everybody, and we all have a lot going on. And we really thank you for choosing to spend the afternoon with us. In order to deliver the best webinar that we can for you, we've put together a few guidelines. First, in your Zoom window, which should be on your screen in front of you, you'll see that there is a button in the top right corner. I don't know which way it is for you, that says Speaker View. Please click on that. This will keep the speaker in focus for the duration of the meeting. Your microphone and video have also been turned off. If you could also leave those turned off this entire time, that would be great, just so that we're not distracting anybody and we can all pay attention to the speaker, who's great. Also, closed captioning is being provided during this webinar. If you'd like to change the size of the text, please click on the arrow that's next to the video camera icon in the bottom bar of your Zoom window. Next, you'll click on accessibility, and that is where you can adjust the text size using a sliding scale. I will be on call throughout the entire meeting today to help you out with any technical questions you may have. Please feel free to email me if you need any clarification or Zoom support. I'll be adding my email and also my cell number to the chat if that's easier for you. You can shoot me a text or give me a ring, either work. In addition to this, the webinar is being recorded. So following the session, we'll be posting it on our social media channels and our website. Feel free to share it with your friends, family, and colleagues as well. I hope that you enjoy the webinar today. And again, thank you so much for joining us. I'll now be introducing you to Christine Hughes, who is Burke Neurological Institute's Vice President of Institutional Advancement. Thank you. You just have to unmute yourself. Sorry.
Thanks, Sophia. Our speaker for today is Dr. Vibhu Sawhney, and he will be talking to us about stem cells and their potential to repair the nervous system. As I am sure you know, this is a very important topic, especially now when there are more than 600 chronic neurological disabilities impacting approximately 1 billion people worldwide. Dr. Sawhney received his degree in dental surgery from the University of Mumbai and his doctorate in neuroscience from Northwestern University. Dr. Sawhney has a specific interest in cortical spinal neuron biology, nerve cell projections that travel from the cerebral cortex region of the brain down to the spinal cord, due to its direct clinical significance for recovery after spinal cord injury, stroke, traumatic brain injury, cerebral palsy, as well as neurodegenerative diseases such as motor neuron disease, ALS. He is a cellular and molecular neuroscientist with a strong background in training in regenerative and rehabilitative neuroscience. His research interests lie in identifying developmental signals that control specificity and circuit development and have a direct impact on nervous system repair. In his PhD work with Professor Kessler at Northwestern University, Dr. Sawhney investigated developmental mechanisms and responses of early stage nerve cells following spinal cord injury. During his postdoctoral work with Professor Maklis at Harvard University, Dr. Sawhney established new anatomical, molecular, and genetic approaches to identify novel genes that control the development of specific corticospinal connectivity. This established new genes for the first time in the field that control how these long projections from the brain to the spinal cord are established. After leaving Harvard University, Dr. Sawhney joined the Burke Neurological Institute in 2018 as the Director, Laboratory of Cell-Fate Specification and Circuit Development, and he is also an assistant professor at the File, Brain, and Mind Research Institute at Weill Cornell Medicine. Welcome, Dr. Sawhney.
Great, thanks, Christine. Let me just share my screen. Okay. All right, can everyone see my screen and hear me okay? All right.
Yes, perfect.
All right, well, thanks, Christine, for that introduction and for putting this together. Thank you for all the participants for showing up. As Christine said, I know there's a lot going on in the world right now. And so thank you for taking the time to attend this webinar today. I thought in light of the recent events that have basically been happening over the past week or so, I would be remiss if I didn't speak a little bit about, because this is something that I think I've been reflecting on personally, and I think we all are, just about building our communities in a way so that every person, regardless of race, color, orientation, gender identity, has the equal opportunity across the board. And I just want to say, I know I'm just saying this because I've been thinking about it, and I know all of us have been thinking about it, and we just had a town hall yesterday at Cornell talking about this. And I think if we start reflecting on our own roles in our own community, I think it'll start to reflect in our society as a whole. So I just wanted to say that. And with that, I'm just going to move forward with the topic at hand for today, and that is about stem cells for treating the nervous system. And I think, as Christine said, my lab or my research interests are not exactly stem cell specific. I'm more interested, or at least my lab is more interested, in how do the neural circuits establish themselves during development, how do we get specific connections, and how can we basically reestablish them for recovery after injury. But I have a peripheral interest in stem cell biology, and so hopefully I'll be able to communicate some of them to you today. And I promise if I don't have the specific questions or answers to your questions today, I'll get back to you with answers if need be. All right, so just to go over what we're going to discuss today as a broad outline of my talk, I'm going to first describe what do we mean by stem cells, because this is a question I think that comes up quite a bit in the lay public. What are stem cells exactly? Where do these reside in the body? Because people hear terms like umbilical cord stem cells, skin stem cells, or stem cells in a petri dish. So what exactly are they and where do they reside? And then I'm going to dive into what evidence do we have currently with the biological sort of understanding that we have of how can these be used to treat the nervous system. I'm then going to talk a little bit about how can stem cells in a dish, even without putting them into people, help treat the nervous system. And then finally, in the era of the COVID-19 pandemic, there has been talk about the use of stem cells for treating COVID-19, and I'm just going to briefly touch upon what the science behind it is and is there any merit to it. All right, so let's dive right in. So what is a stem cell? So a stem cell is basically any cell in the body which has two main defining characteristics. And one of them is this idea. So here what you're looking at in this cartoon are basically cells which are residing actually in a dish. And what these cells can do is they can divide and make new copies of themselves. So essentially, these cells are programmed to not only give rise to other cells, but they can make multiple copies of themselves. And this is a property that's called self-renewal. So they can keep on making multiple copies of themselves. Different kinds of stem cells can do this to varying degrees, but this is one defining characteristic. And the other defining characteristic is that if you take these stem cells, they can differentiate into different kinds of cells. And by that, what I mean is that they can make cells that are different from themselves. And this is defined as differentiation potential. So which basically means that this stem cell, and in this cartoon, this is an embryonic stem cell, and I'll describe what that means in a minute. But this stem cell can actually give rise. Can you guys see my pointer? I just want to make sure that that's the case.
Yes.
Okay. So this stem cell can give rise to nerve cells, cardiac cells or cells of the heart, blood cells, liver cells, and so on and so forth. And different stem cells actually have different degrees of differentiation potential, and I'll describe what that means next. So if you now go back in development, when an organism is first generated, right? When the sperm fuses with the egg, you get what's called a zygote. So this is the one cell, the first cell that basically is going to give rise to the entire organism. So whether this is a mouse or even a human, this is going to give rise to that entire organism once it's implanted inside the uterus, for instance. This cell will then divide and give rise to an embryo. And in this schematic, basically, there's an outer layer of cells, and you can see that there are these blue cells that are sitting inside. I'll come to that in a minute, but from this single cell, you get multiple rounds of cell division. So this will make multiple copies of itself, and you get an embryo. And then further down the road, this embryo is going to have different cells that get specialized even further into what can give rise to what are known as the germ layers. So this happens, for instance, by about the second week in the life of a human embryo. You get specified cells that are going to give rise to, on one end of the spectrum, the inner linings of our body, so the cells of your gut, cells in your lung, your liver. The outermost layer, so the skin of your body, is going to come from a different set of cells, and then all the stuff that's in between. So you'll have cells that are going to give rise to your heart, your skeletal muscle, your kidneys, and so on and so forth, your connective tissue, tendons. Now, what this diagram is hopefully showing you is you start from a cell that can give rise to the whole organism. You then get a bunch of cells that are a little more limited. So these cells now, if you basically take them out and put them inside, say, the uterine wall, they won't be able to give rise to a whole embryo or a whole animal. And then now you have cells that are even more specialized to give rise to distinct, specific kinds of tissues in the body. So there are genes that are going to be directing them to become more specialized. And if you think about this, this sort of makes sense, right? Because if you have some cells that are going to make, say, your gut, you don't want them making proteins or molecules that a nerve cell needs to function or your heart cell needs to function. So you need to specialize these cells to make different tissues in your body. So if you go down this axis in time, and actually if you go down this axis in terms of cellular space, there are going to be cells that are able to basically make every single cell type in the body. So these are defined as totipotent. They're potent. They're able to make, their potency is to be able to make every single cell type that makes an animal. You go a little further down, and then you get what are called pluripotent stem cells, and those are the blue cells that are described here. These cells can actually differentiate into every single cell of this lineage, so you can get these cells to differentiate either into the gut, the skin, or the heart. And the cartoon that I showed you just before this, those embryonic stem cells, those would be examples of these pluripotent stem cells. You can get nerve cells from them, you can get blood cells from them, you can get cardiac cells. Now, if you go further down this road, you'll get more restricted stem cells that are basically residing in different parts of this tissue. So you get what are called multipotent stem cells. And another word that you probably sometimes see in the field that describes them are progenitor cells. What this tells you is that they're a little more limited in what they can make. So normally, during development, if you have, for instance, a stem cell that's going to give rise to the skin, it can give rise to multiple tissues within the skin, but it's not going to give you, say, cells of the liver or cells of the heart. So our region of interest, the nervous system, basically comes from this part of the trajectory. So you go from the zygote to the embryo, and then from this part of the embryo called the ectoderm. And now I'm just going to walk you through what these sort of definitions then mean for how do you get a nerve cell in the brain or in the spinal cord. All right, so this is where we started from. We started from the totipotent zygote. We then developed an embryo. And then if you are wanting to make the nervous system, we further specified this, so you have what's called the ectoderm. This is either going to give rise to the skin, or the other arm of this is going to give rise to neural tissue. And what I want to emphasize is when I say these kinds of sort of bifurcation arrows, there are genes that are going to drive the program one way or the other. So there are genes that are going to specifically get you nervous system tissue as opposed to skin. All right, now what happens once you specify this neural tissue? Well, the first thing you're going to make, and this is all during normal development, so this is happening in the human brain as it's developing inside the embryo, you'll get a bunch of these cells, so these sort of blue-green colored cells that you see here. This is sort of the lining of the brain. This is the inside of the brain, and that'll be the outside. You don't need to worry about these specific terminologies. But what you can see is that this one cell, for instance, will divide. That's what this sort of schematic here is schematizing. And then from 1, you sort of go to 2. And then this process is going to keep on happening for a sustained period of time so that you generate multiple copies of these cells. So this was the self-renewal that I was telling you about. Then over time, if you want to make the nervous system, these cells are going to divide and they're going to give rise to, from these cyan-colored cells, you're going to get these purple-colored neurons in this cartoon. So you're not only now maintaining these stem cells, they're making more copies of themselves, but what you can see is they're starting to divide and they're giving rise to nerve cells that will go on to become a part of the brain. Now, this is going to become more and more complex. You don't need to go into the details of this because obviously our brains are super complex. So you're going to get multiple different kinds of nerve cells that are going to be generated. But what I want you to notice is that these stem cells that you started from at the very beginning here, they're sort of being maintained. They're giving rise to other kinds of stem cells that are also then going to go ahead and differentiate and give rise to different kinds of neurons. And then finally, you get later on in development, they've generated extremely complex cell types. Some subsets of these are maintained as opposed to the large numbers here. Their numbers have declined. And now you've generated a whole host of different kinds of neurons in the brain. So the point that I want to illustrate here is that normally during development, when you've started from this sort of process, you have normally stem cells that are residing in the developing human brain whose job is to make multiple copies of themselves and then generate all the different cell types that are going to make up the human brain. These cells are not going to make cells at the heart. They're not going to make cells of the skin. They're specifically designed to make cells that are going to give rise to the nervous system. So these are neural stem cells. They're specified during development, and through time, they're going to basically get progressively restricted in what they can become or what they can differentiate into. And so a lot of work has, for instance, gone into looking at what this normally looks like in the developing human brain. And this is work from Arnold Kriegstein's lab. And so here what you're looking at is actually a slice of the developing human cerebral cortex, which is the outermost part of the brain. Each one of these green blobs that you see is actually a single neural stem cell. This is about in the first trimester of gestation. And I'm going to play this movie. And first, what I want you to just see is that these cells, these cells are going to be highly dynamic. So I'm just going to first play this movie, and you can see that these cells are very, very dynamic. They're moving up and down, back and forth. And let me just pause this right here. What I want you to now start focusing on is this cell right there. I'm going to play this movie. Sorry, so you're going to see this cell. Let me back that up. This cell, which is going to now pop down here. You're going to see an arrow forms. Yeah. And now focus on this cell. It splits into two. One copy of it is going to go up, and the other copy is still staying here. I'm just going to play this part again. So this is happening live inside your human embryo. There's the cell. There it splits into, one copy goes up, the other copy is still staying close to where it was supposed to be. So the point that I want to make here is that these are stem cells that are normally present in the developing human brain. These are not inside a dish, and their job is to now divide, make more copies of themselves, and give rise to all the neurons, all the nerve cells that are going to be a part of the developing human brain, and eventually in the adult. Now, once development is complete, so just to go back to that sort of definition, the stem cells that are described to you that are designed to give rise to nerve cells, those would be examples of these multipotent progenitors. They're not going to give rise to any other lineage, but they are still actively dividing, and they're still sitting in the brain giving rise to new nerve cells. All right, so once development is complete, within each one of these tissues, what is left over is that you'll still have some stem cells, and these are adult stem cells. So they are no longer giving rise to developing tissues. The tissues have developed, you have matured blood, skin, the brain is developed, but there are some populations that are then left behind in the adult body. And so classic examples of these are the hematopoietic stem cells that throughout your life are going to give rise to new blood cells. You have stem cells in your skin, mesenchymal stem cells, and I'm just bringing up this point because a lot of these stem cells you would see out there in the literature that are used in clinical trials, especially for treating the nervous system, and I'll come back to that point in a bit. And then even in if you want to now go back in time, these adult stem cells are going to be far more restricted in their potential compared to, for instance, what these embryonic cells were able to produce. So if you go back, I told you these embryonic cells, for instance, can give rise to all the lineages of the body. These cells are going to be far more specific. So a neural stem cell is just going to give rise to the nervous system. Hematopoietic stem cells normally will just give rise to blood cells. And so the way people have now originally gone about isolating stem cells was to take these embryos early on in development and then put them in a dish and grow what are called embryonic stem cells. And then these stem cells could then be differentiated into each one of these lineages. But another sort of idea about repairing the brain came from the fact that you have these neural stem cells that also reside in the adult. And this led to the idea that potentially we can investigate and use these neural stem cells. Instead of taking them out, putting them in a dish, can we actually use them to repair the nervous system from within itself? And so the classic way of studying this was you take these cells out, throw them in culture, throw them in a dish, and you give them sort of growth factors, essentially any molecule that can promote them to divide. And these were a lot of the experiments that I actually did during my PhD. So just to give you a context, if you basically take these cells and just because they can divide and make more copies of themselves in culture, you get what are called these neurospheres, or these balls of cells. And just to give you an idea, you see all these other dots in this picture. Each one of those is actually a cell that was also put in, but they did not have the capacity to divide. So this big sphere that you see here actually originated from one of these cells that divided over and over and over again, and it gave us this neurosphere. And then what people have classically done is taken this neurosphere, plated it out, and then asked, can it give rise to the three sort of principal cell types that you see in the nervous system? So you have nerve cells or neurons, and then you get glial cells, astrocytes or oligodendrocytes. These are glial cells. These are the nerve cells that fire those electrical impulses that allow us to do all the things that we do. And classically, although this, some of the glial biologists will kill me for this, but classically the glial cells have been thought of as support cells of the nervous system. Now, the interesting thing about this is that in the adult, once development is complete, it turns out that even though these adult cells can give rise to neurons in a dish, in vivo, there's now evidence that that actually doesn't happen. So these neural stem cells that are residing in the adult brain classically just give rise to these glial cells, and they don't make new neurons. So that once you've lost neurons due to injury or disease, say in stroke or in neurodegeneration, you don't have the ability to take these stem cells and direct them, to drive them to become new neurons. And so a lot of the efforts, for instance, have been towards, can we actually recapitulate some of this process in the adult brain to use these stem cells to drive repair from within? And this is some of the strategies that are schematized here. I'm not going to go into the details of this, but just to tell you that some of the thoughts have been, and there is evidence for this from work that has been done in animals. So work from Magdalena Gutz in Germany, for instance, where they've taken these stem cells that reside in the adult that normally don't give rise to new neurons, and they can actually take genes and program them and differentiate them into new neurons inside the cerebral cortex, inside the highest part of the brain. And so this is an interesting idea, where the idea would be that these adult stem cells that are sitting inside the brain, you don't have to take them out, you can actually manipulate them and drive them and trigger them to becoming new neurons and repairing the brain, the damaged brain from within. All right, so just some take-home points that I want to emphasize before I move on. So the question that I started with, what are stem cells? So the answer is that the stem cells are self-renewing cells. These are cells that can make more copies of themselves, and they have varying degrees of differentiation potential. So you have cells that can give rise to many, many different kinds of tissues in the body, and then within each tissue, you have cells that give rise to more specialized kinds of cell types. You have neural stem cells, blood stem cells, and so on. These, so that's what I just told you. They reside in multiple tissues during development, and then within each tissue, they give rise to multiple different cell types. These stem cells are also present in the adult, although their numbers are much smaller than what was present during development, and their potential is a little more limited. So for instance, the neural stem cells that are present in the adult brain are not capable, at least by themselves, to give rise to new neurons, new nerve cells. They'll give rise to glia. And so the idea has now been that these adult stem cells that reside in specific niches and specific regions of the brain, they represent a potential source for nervous system repair. All right, so going back to that first diagram that I showed you, where along the axis of time, you start with a totipotent cell, then you get a more differentiated cell, and then you get an even more differentiated stem cell. And so this arrow is sort of showing you that their differentiation potential is high. You can get many, many different cell types here, and then fewer different kinds of cell types the further down you go. And so the idea here was you can take these early embryos and get embryonic stem cells. Work that was then done by Shinya Yamanaka about 15 years ago or so showed that actually this is not just one direction in which this process can proceed. So in fact, the reverse is actually true. So you can actually now force, for instance, a skin cell to go back in time in development by forced expression of specific genes, and you can take these skin cells and revert them back to cells that almost look like embryonic stem cells. And that's schematized here. So imagine what this means now is that if you are a patient of whatever disease, you can pick your favorite disease of interest, you can take a skin biopsy, for instance, you can grow these skin cells in a dish, you can add the genes here that are listed. The names aren't important, but essentially what that does is takes these skin cells and reverts them back to cells that now almost look like these embryonic stem cells. And these cells are called induced pluripotent stem cells. And the advantage here, for a whole host of ethical reasons, for instance, if there were ethical considerations when it comes to embryonic stem cells, because you do have to sort of destroy human embryos to get human ES cells. Now, you don't actually have to do that. You can take stem cells, or you can make stem cells out of adult cells, and these cells would in fact even be patient specific. So we can make stem cells that would be specifically similar or identical in their genetic makeup to the donor of origin, right? So each patient can, for instance, have stem cells that are that are identical to their own genetic background. So that sort of takes away things when you considerations of things like, if you were to transplant these, you don't have to think about immunosuppression because there's no graft versus host response kinds of problems that might occur. But the other point that I want to make with this is, as ethically challenging as embryonic stem cell research was, this finding about induced pluripotent stem cells would not have been possible had it not been for decades of embryonic stem cell research work. So the fact that that this red arrow is possible was actually driven by detailed, painstaking research, where researchers were able to identify what makes these embryonic stem cells tick and what makes them differentiate into these different lineages. And it was taking actually advantage of that kind of biology that we were able to then reverse engineer development and sort of go back in time. And so, as ethically controversial as that was, it actually spawned the field of induced pluripotent stem cells, or iPS cells, which otherwise would not have been possible. So I think that's one thing that we have to keep in mind about science, that sometimes, in fact, the projected outcomes of the research that we're doing today are hard to predict, because 15 years ago, when Shinya Yamanaka, for instance, first presented his findings about this very biology, the majority of the world did not believe him, and it was met with intense skepticism. But now this opens up a whole host of options that are available to us, right? So if you can now make iPS cells that are in fact patient-specific, the options now become tremendous. So you can take these patient-specific iPS cells, and on the one hand, what you can do is you can differentiate them into specialized cells of whatever disease you're interested in: cardiac cells, so heart cells, skeletal muscle cells, neurons. You can screen them with different drugs. You can then develop new drugs, and then you can validate them by actually treating patients. So you don't actually have to do large-scale trials. You can screen hundreds of thousands of drugs on these cells in a dish before you even go into a patient. And on the other side of the spectrum, you can take these cells, potentially, for example, if you have a gene deficit or a genetic disorder, you can imagine that you can actually correct this disorder using gene alteration technologies such as CRISPR, have repaired iPS cells, and then put back healthy specialized cells that you can transplant them back into the body, into the patient. As exciting and as promising as this has been, there's been a lot of work that's been done in this regard. The successes from this still remain limited, but I think it's just because we haven't been able to get the right kind of differentiation protocols, et cetera, established. But I think this still remains an extremely powerful and useful technology, and I'll come back to this towards the second half of my talk when I talk about their use for the nervous system. But now I'm going to switch gears a little bit and talk about what we know, given this background now about stem cells, where they come from and how we can make them both in a dish and in the body. What is the evidence that we have for treating or using them to treat the injured or the damaged nervous system? So one classic sort of idea is cell replacement. So this is whether it's a patient of neurodegeneration where neurons are lost, or in a patient of stroke where, again, neurons are lost. The idea is, well, let's take these stem cells, differentiate them into the neurons that we want, and replace the damaged or lost neurons. Well, this, it turns out, is a lot more complicated than I just said. But for this, what I'm going to show you is one example where people have actually done this successfully. Now, the classical sort of way in which people have gone about doing this very idea, or executing this very idea, is in two ways. They've either taken embryonic stem cells or taken iPS cells from both healthy donors and patients, and then what they've done is differentiated them into progenitor cells that are going to give rise to the neurons of our interest. And I'm going to give you one such example where I think the biology has been done right, and that is in the instance of Parkinson's disease. So Parkinson's disease is a neurodegenerative disorder in which there are these specific kinds of neurons that sit in this part of the brain called the midbrain. You can see them, they're black in color. They're called substantia nigra. So they make a neurotransmitter called dopamine. And you can see that in a Parkinson's patient, you lose these very, very precisely lose these specific populations of neurons. that are in the midbrain. They're responsible for a whole host of motor coordination and motor ability. So Parkinson's patients, for instance, and you've seen this disease was made famous, for instance, by Michael J. Fox. I like to point this out because growing up in India, there were few things that were, of American origin that I actually watched growing up, but Back to the Future movies were definitely something that I was aware of. So when he was diagnosed with Parkinson's disease, it was something that even I, sitting in Mumbai, 10,000 miles away, could relate to. But Parkinson's, for instance, because of the loss of these very specific populations of neurons, you get basically difficulty in movement, you get those tremors that you've classically seen. And so the idea was that this would be a good test of the ability for stem cells to actually replace this one type of neuron that is lost. Because as far as complexity of disease is concerned, we know the type of neuron that's lost. So let's just make the right kind and put it back in. Turns out much, much easier said than done. Because as I'll show you next, if you went back about 15 years, everybody who was working with embryonic stem cells wanted to make this kind of neuron in a dish. And they had pretty beautiful looking pictures where they could show that these neurons, shown here in green, so that this lower panel is what I want you to focus your attention on. These are cells that have been differentiated from embryonic stem cells to become the dopaminergic neurons. And the way we know they're dopaminergic neurons is because they make this enzyme called tyrosine hydroxylase, shown here in green. But it turns out that you can't just make a generic dopaminergic neuron. The dopaminergic neurons that are lost in Parkinson's disease are a very, very specific kind of dopaminergic neuron. And for that, these neurons need to also make two other proteins, FOXA2 and NER1. The gene names, again, aren't important, but you can see, so if you stain for those in red or blue, the conventional approaches failed terribly at making the right kind of neuron. And it wasn't until Lorenz Studer's lab in New York actually figured out what was the correct cocktail of getting the right kind of neuron that progress could be made. So here you can see that they've basically taken conventional ways of making dopaminergic neurons. And you can see you can get dopaminergic neurons, but they're definitely not of the right kind because they're not making these two other proteins that the specific neurons that are lost in Parkinson's should be making. So when they figured out how to do this, they could actually robustly, efficiently make these neurons in a dish. And then they went ahead and tested whether these neurons could actually integrate into the host. So they used these in mouse models of Parkinson's disease. And what I want you to focus on is actually this panel right here. So these are the grafted neurons. So these are human neurons that have been grafted into the mouse. And in this, what they're doing is they're testing the behavior of these mouse models of Parkinson's. So on this plot, the higher the score on the y-axis, the worse the animal is. And if you take these kinds of neurons that are dopaminergic, but they're not of the right kind, and you put them into the brain, you see that they actually might get better, but then over a while, they actually start getting worse. So there's actually no functional recovery when it comes to analysis using this. But if you put the right kind of neuron, the neuron that they've now, over decades of work, been able to establish, you can see that they actually improve tremendously. These neurons graft, and they actually promote meaningful recovery that is sustained and long-term. So they went ahead and also did the next step experiments. They put these cells in rat models of Parkinson's and even in the monkey. And the reason why I bring this up is this is building on a ton of developmental biology research that involved differentiation of stem cells, mouse genetics, cultures, co-culture experiments, and that's how they were able to figure out how to make the right kind of neurons. So even though the idea was very, very simple, right? Let's just make the correct kind of dopaminergic neuron in the midbrain. Getting to that point was very, very challenging. But now that they've done it the right way, the next step now is they're trying and testing this out with human patients. So here, I'll show you in the next slide, there are 4 different clinical trials that are currently underway, and they're taking different sort of iterations of this process. So in one set of studies, so these are being conducted in Japan, they're taking basically normal people, they're generating iPS cells from their skin. So these are those cells that are gonna come from skin biopsies, for instance. and they're going to differentiate them into the dopaminergic progenitors. So using the protocol that I just described to you that gets you the right kind of cell. Alternatively, you can take embryonic stem cells, differentiate those into progenitors, or you can take patients that have Parkinson's, make iPS cells from them and differentiate those into dopaminergic progenitors. And the idea here on the left-hand side would be that you will match these donors by the HLA donor matching program. So this is sort of similar to what you would do if anyone was getting, for instance, a bone marrow transplant. And the idea here would be that because it's the patient's own cells, when you put them back in, you won't even need immunosuppression. And so these are sort of the 4 different centers. So there's the trial that's going on in Europe, there's the New York STEM trial, there's the one in Japan. And what I want you all to see is that there's either the use of embryonic stem cells, iPS cells, and even autologous iPS cells. I'm not going to go into the details of this because a lot of this is still ongoing, and we don't have the results from it yet. But even with all of this biology that's been teased out and laid out and nailed down to perfection, in my opinion, there's still a host of considerations that we have to think about. And so one of them is cell testing. So once you've differentiated these progenitors in a dish, do you every single time, for instance, take these differentiated cells, put them into an animal to screen to see are they still efficacious before you actually put them into people? And now if you go back and sort of think about that, right? So here you can sort of mass produce these cells. So you can probably test different batches of them, put them into animals and see, well, are they actually behaving the way they're supposed to before you go into people? But now imagine doing that for this side, where you're making iPS cells from each patient so that they won't be immune rejected. Doing something like that would be tremendously cost prohibitive. But these are some of the considerations that international sort of research groups are coming together and asking to see what's the best way forward. So that's one thing. Some of the other things that we have to consider is screening for mutations, because you're growing these cells in a dish. They're dividing uncontrollably at times. So maybe as they're dividing in culture, they develop mutations in their genes, in their genome, that might actually make them tumorigenic. So even though you're trying to differentiate them into dopaminergic neurons, once you put them inside people, they might actually turn into tumors. So you can actually screen or sequence their genomes to make sure that at least some of the tumor-causing cancer mutations do not exist. But then the question is, where do you stop? Do you stop at just looking for cancer-causing mutations, or do you look for other genetic variants? So now we have next-generation sequencing. Do we sequence the entire genome? Do we sequence specific loci? Do we sequence those parts of the genome that we know are going to give us more susceptibility towards, say, other kinds of disorders? And these are all considerations that I think we need to think about as a field, as we even take well-researched biology and bring it to human clinical trials. So some of the other sort of avenues by which stem cells can be used to treat the injured or damaged nervous system. So apart from cell replacement, which I just told you, there's a whole host of other ways in which stem cells have been purported they can actually produce recovery. And this is because a lot of times people have taken stem cells and put them in animal models and experiment models of stroke or spinal cord injury. And some of these sort of prevailing ideas is that they're not actually replacing the damaged cells, but what they're doing is, for instance, providing trophic support. So they can actually, so you've damaged the nervous system, cells have been damaged, cells have been lost, neurons are gone. But then whatever's left behind, these stem cells can actually provide trophic support so that those additional sort of secondary events of cell death, et cetera, do not happen. You can also think that these stem cells can differentiate into immature neurons and provide new relays that can compensate for the loss of the neural circuits. So some neurons have been damaged. You basically are building new circuits that can provide for some recovery. And then this other point is immune modulation. And I'm just going to show you a slide about what I mean about that. So in the normal nervous system, the blood cells, which are shown here on the top, do not gain entry into the nervous system. So on this side, so this is sort of blood vessel and all of this is brain. And that happens, and this sort of exclusion happens because of the formation of what's called the blood-brain barrier. What this basically means is blood cells do not gain access or into the, blood cells do not gain access into the brain or the nervous system. And this happens because there are glial cells called astrocytes. And so these toadstool-shaped sort of endings that you see here, they're responsible for maintaining this barrier. They're very, very specialized cells that can sort of control what gets into the nervous system and what doesn't. Now, when you have neurodegeneration, so for instance, this healthy looking neuron in the healthy brain is now degenerated, you see that this blood-brain barrier breaks down. And now, so immune cells from the circulating blood circulating system can gain access into the nervous system, and they can actually amplify whatever inflammation is going on. So one thought, for instance, is you can take glial cell transplants and you can help seal this blood-brain barrier again, preventing extra infiltration. You can also modify the extracellular sort of components. So there's evidence for this in Alzheimer's disease, but the idea here is that maybe these stem cells sort of engineer the extracellular environment around these neurons that prevents them from dying and helps them sort of survive and thrive. And these are sort of already instances that I described to you, where the idea is they actually promote the survival of these neurons either directly through trophic factors or they can replace them. Okay, so going back to that initial slide that I showed you, stem cell-based treatments, so the ones for Parkinson's disease that I described to you, either used embryonic stem cells or iPS cells. Some of the other trials that have been done for things like stroke and spinal cord injury have used adult stem cells, and more classically, these hematopoietic stem cells, so blood stem cells, and mesenchymal stem cells. And the idea is that you can differentiate them either into nerve cells or actually just transplant them with the idea that they can provide some goodies or trophic support for the cells that are lost or damaged. This is not because they're better suited or that they're better adapted for this purpose. It's more because of ease of access, frankly, because people have been able to get these cells more easily from patients. So getting the hematopoietic stem cells out of the bone marrow is an established procedure, as opposed to, for instance, trying to get neural stem cells out of the brain tissue. And the idea here is that if you think about for things something like stroke, you can take these cells, if they're iPS or ESL derived, differentiate them into the right kind of glial cell or neuron and put them into the stroke brain. Now, the idea that I want to emphasize here is that these treatments are for tissue regeneration. So they're very different from treatments for stroke that actually are aimed at the acute phases of stroke. So for instance, acute treatments of stroke would be something that are aimed at reducing tissue damage in the very, very early stages. These therapies are designed with the idea that, okay, damage has happened. How can we make the damage system better? And there's a whole host of thought processes of how they can do that. The only point that I want to make is even in this schematic, and a lot of these schematics that you will see in the field and out in the literature, people draw these arrows from iPS to glia or neurons of the right kind. And like I showed you with the dopaminergic neuron, where they were trying to get the right kind of neuron, it's a very, very complex process, and it's not as easy as perhaps some of these schematics would lead you to believe. But regardless, there have been potential targets that these stem cells have been sort of attributed to perform. I'm not going to go through all the details of this, but the idea is it's not replacing the damaged cells, it's sort of augmenting whatever function potentially was left behind by the circuits that weren't damaged by the initial assault, but are potentially vulnerable to secondary rounds of assault. Stem cell treatments, for instance, for tissue regeneration after spinal cord injury. So in addition to the ideas that I just mentioned earlier, in spinal cord injury, one of the things that's happened is that the connection between the brain and the spinal cord, these long connections that are coming down, in fact, these are some of the connections that my lab studies, have been damaged. So the idea is that potentially these stem cells can be put into the lesion site and they can promote growth of these axons, these long connections, and in fact also provide new relays. So maybe these long connections synapse or make connections with these grafts, and that can actually help augment functional recovery. Now, all of this said, I think I could detail here a whole host of different clinical trials that are going on with stem cell treatments for different kinds of diseases, but I'm not going to do that just because I think there's multiple concerns, for instance, with the what should we be concerned about moving forward with this? So one of the things is the cell source. How reliable is it? How confident are you that the cell source that you're working with is the correct cell type, for instance? And then when we're analyzing these in clinical trials, it's hard to decipher how much of the recovery that you see is actually because of your grafts, or how much of this is just something that might have otherwise occurred spontaneously. This is something that has occurred more recently with stem cell tourism, where patients have actually gone abroad to get stem cell or purported stem cell treatments that have not been vetted, basically where the cell source was not carefully sort of characterized or identified, and they've ended up with complications. And I think there's a real potential for worsening outcomes. And some of this is, as I just mentioned, you can get tumors, you can get infections. There can be inflammatory responses. And basically, you can also make abnormal circuits. You can have neurons in the wrong place in the wrong spot, and they can make a bad situation far worse. And here I'm just showing you one instance, and you can actually even look at this case study. This was a 62-year-old man who had legal blindness because of optic neuropathy, who went to China to get a stem cell transplant, not vetted, and he basically then started developing bilateral weakness in the legs, pain, and that was because he started getting compression of his spinal cord. And you can see this intraoperatively, this is actually growth that has come from those stem cells, and they've done sequencing to make sure that this was not his own tissue. Those stem cells have actually formed tumors inside his spinal canal. So even so, he was legally blind. He's now gone and gotten stem cell treatments, and that have actually made his condition far worse because he's now having walking problems. So I think we have to be very careful about what kind of treatments we're putting into people, because I think they have to be very carefully vetted, because the potential for worsening outcomes is far worse, is really significant. So to close this part of the talk, how can we use stem cell transplants to treat the nervous system? So I've given you one example where I think they're doing it the right way with cell replacement in Parkinson's. And then there's the idea that you can actually do these other mechanisms to promote function out of whatever's not damaged from the initial assault. Some of the concerns, if you don't have the right cell type, it'll affect efficacy. If you don't have a complete understanding of what cell type you're working with, you will not be successful. And there's a real potential for worsening outcomes if you don't get perfectly controlled trials. All right, so I'm going to switch gears now in the last sort of 5 minutes of my talk and talk about how can stem cells in a dish be used to treat the nervous system. And I already gave you this idea of you can make these specialized cells. So imagine these are nerve cells. You can screen them with different drugs, get target validation, and then treat patients. And in fact, some of this work is actually going on right here at Berkeley. So Gary Gibson's lab is looking at Alzheimer's disease, and they've taken iPS-derived neurons they can put in a dish, and they're actually looking at some of the abnormalities in glucose metabolism between patients who have Alzheimer's versus control neurons. And I'm not going to go into the details of this, but the idea is that you can actually now start to ask very specific questions of the kinds of neurons that are specifically critical to the disease of your interest. But I think there's another interesting idea here, and I think that's exemplified here. So in all of our labs, the classic sort of mouse model is what's being used to investigate disease and investigate the nervous system. But through evolution, there's been a tremendous expansion of the brain. So the mouse, for instance, has about 71 million neurons. We have 1,000 times more neurons in the human brain. These neurons are far more complex. They make far more complex connections. And just think of what we can use our brains to do. We're not mice, so we're far more intelligent, at least some of us are. And they can actually, so the human brain, for instance, is not just a big mouse brain. So how can we get to the kind of complexity that is present in the human brain that is robustly lacking in the mouse brain? And especially if we have to treat disorders which are very specific to humans, Alzheimer's disease. I mean, I don't know if there's a mouse in the wild that has Alzheimer's disease or a mouse in the wild that has schizophrenia. That would be kind of freaky. But the idea is that a lot of these human-specific disorders will need human-specific tests to be able to identify what's specifically wrong. And so as opposed to all the diagrams that I've shown you so far, where stem cells were plated and you could get neurons in a dish, some of the ideas that were actually, that originated a little over a decade ago, were that you can use stem cells in a dish to create complex 3D structures. And this is what pioneering work that was done by, in my opinion, Yoshiki Sasai in Japan, the late Yoshiki Sasai. And what he was able to do was take stem cells, and I'm just gonna play you this movie, and what you're gonna see for here is you're gonna start with, and these are mouse embryonic stem cells. And what they were able to make, so here's a ball of mouse embryonic stem cells. And you see how, I'm not gonna go into the details of what they actually did, but you see how here, these stem cells are starting to produce these protrusions. And what they're going to try and show you, what they're going to show you next is that they are going to form little tiny retinas in a dish, a complex 3D structure that is being made. So this is going to go on. You see it sort of protrudes out, and then see what happens next. So this is one of those protrusions, 6 days in culture. Now over the next 2 days, you see how it invaginates. So it basically makes this little cup. And you're going to see another example of that here. So again, keep focusing on this or here, you can see it will start to protrude inside and invaginate. And now if this were a normal mouse eye, you'll have the lens here, and this is the neural retina. And you can see that this is stem cells that are basically sitting inside a dish, and they're producing these really complex 3D structures. And if you look at the structure, you can actually see, so this is what it looks like in the dish, and this is what a normal mouse retina looks like. See how the retina, all this red stuff, see how closely it matches what you would normally develop inside the mouse embryo. And that led to the idea that can you actually make cerebral cortex, or basically different organoids, mini organs in a dish using embryonic stem cells. And this is one such example of basically the cerebral cortex that was pioneered in Jürgen Noblich's lab in Austria. And now what people are doing is taking these organoids, and now you can think of modeling diseases like microcephaly or any specific human-specific disorder. You're actually working with human neurons inside a dish. And now the idea is that you can basically produce some of the complexity that a mouse brain simply cannot. All right, so just to close this part, how can stem cells in a dish inform treatments? So this is informing treatments without actually taking the stem cells and putting them inside people. We can produce neurons for drug testing. We can generate complex 3D structures, and the idea is that this gets you better access for further investigation. You can model complexity of the human brain, which is potentially absolutely critical if you want to model human disease. All right, just a final closing thought. Because we're in the era of the COVID-19 pandemic, there have been stem cell treatments that have been purported for treating COVID-19, and I've just co-opted the slide for those of you who attended Dr. Rattan's excellent webinar, I think it was 2 months ago, where basically the SARS virus can gain access into the lung epithelium cell and then produce what's called the cytokine storm. You can get vasoconstriction, and you basically get, when people develop these respiratory distress symptoms, that's because the inflammation or the immune cell has kind of gone berserk. And so the idea is that can you modulate that using stem cell treatments? But I would caution, especially in the light of all that I've told you, and I would especially encourage you to read this paper by Lee Turner at the University of Minnesota about how even in the era of a pandemic, we have to exercise caution because there is a significant concern of doing more harm than good. And with that, I would thank you, and I'm happy to take questions.
Dr. Sani, thank you so much. That was a fascinating talk. And I'm just going to remind our audience to please remember to submit your questions. You can submit them via the chat box on the bottom of your screen, and we'll go ahead and start reading. Actually, Bibu, if you can share your screen again and go back to the first movie that you showed us of the embryo cell, brain cell dividing.
Yes.
Just, if you can, give us a little bit more of a context of that. There was a question regarding whether or not that was an organoid or what the origin of that.
Sorry. So I think the question was for this one, right?
Right.
Guessing this movie. No, this is not an organoid. This is actual fetal human tissue, which has been electroporated. So basically what they've done is they've taken DNA that can make these cells green. So every one of these cells has basically been electroporated in, they've used electrical current to drive DNA in, and that's how the cells are visible. So this is not an organoid, but that's an important point. People have actually asked, do the organoids replicate some of these kinds of divisions? And the answer, at least at first pass, appears to be yes, but with some caveats. And I think that the thought is that maybe some of these conditions that that are current conditions of culturing them, the organoids in a dish, may actually be restricting some of the potential cells that you can get from them. Yeah.
Excellent. So we have another question regarding, actually, I think a pretty famous race car driver, Michael Schumacher, who had a serious brain injury, I think back in 2013, 2014. Anyway, I think he was in a induced coma for a while, and now they're talking about doing stem cell surgery to regenerate his nervous system. What are your thoughts on that, and is this possible?
So I think if you can probably surmise from my take on a lot of this is I think that the evidence for it is probably limited at best. I don't know what specific treatments they're trying or testing on him. What I will say is, even though there has been no evidence of clinical efficacy with this, the stem cell trials that have been conducted so far for stroke, at least in 2 of them, there also hasn't been safety concerns. That's not true of all of them. There have been some which have been aborted for safety reasons. So it's possible that for some of those where at least safety concerns didn't arise, that there might be some merit to their use. I fear that in something like traumatic injuries, traumatic brain injuries or stroke, where the complexity of the injury itself, there have been so many different neurons that have been lost. Recapitulating them is obviously a huge, orders of magnitude away from that eventual goal. But even if you put something in which might interfere with potential plasticity, I would be very concerned about putting something in where you now generate, for instance, some abnormal connections, and you turn a traumatic brain injury patient, for instance, into a patient with epilepsy, or in a spinal cord injury patient into a patient with chronic pain. And I think that those are real concerns. I see here there was a question about, is there a summary of available clinical trials? Yes, I can actually, I think I have.
And that was for stroke.
Yes, I actually hid this. I took this slide out, but I can show it to you. And I can even send you this article. So there is a review right here where, and yeah, you can actually look up this review, but I'm happy to send it to Christine, and she can send it along, where this is basically a summary of stem cell treatments that have been done for stroke, and I have a similar such data set or review for spinal cord injury, and I'm happy to send them your way.
Great, and we'll make sure that everyone has access to these slides in this recording. And then there was a question about aphasia, so the neurological impairment of not being able to speak, verbally communicate. What are your thoughts about stem cell therapies in that particular impairment?
So I think there are a couple of things that I think we need to think about before we go down that route. One is, what is the cause of the aphasia? So in other words, what is the neural basis for the aphasia? Is it cognitive? Is it something that is more sort of physical? So like you getting tongue weakness or something. So maybe potentially you might want to get motor neurons that are more specifically going to innervate those muscles of your voice box or your tongue, etc. So I think for the cognitive thing, I think we're way away from that. I would say that for some of those lower hanging fruit, relatively lower hanging fruit of physical causes, there is research that is going on that basically looks at these different cranial nerves, for instance, that innervate different muscles of your face. I'm not totally familiar. It's a little bit outside my real field, but what I would hope is that there we at least have some or a better handle on the developmental biology of those specific neurons, where we could potentially then think about generating those neurons. I haven't seen anything that I would say is compelling evidence for that yet, but yeah, I
guess we'll keep our eyes peeled for anything in the literature. Also, question about, can you use single, double umbilical cord, I guess, as potential stem cells? There is less likely a risk for graft versus host.
Yeah, I think there was a lot of interest in the umbilical cord sort of cell field. I think with the advent of something like induced pluripotent stem cells, I think perhaps the interest in the umbilical cord, as someone who's worked in stem cell labs, I would say that the focus has been now on induced pluripotent stem cells more than the umbilical cord stem cells. In fact, I had some friends of mine who had asked when their kid was born whether they should, save the umbilical cord. My take on it is nothing in the evidence has convinced me that it's totally worth it. And at least as far as the ability of pluripotent stem cells is concerned, with the data that we have today, I think I would say that by and large, they can make what has been shown for back in the day, the umbilical cord stem cells. But I'm happy to look that up and get back to you if there's any if anyone knows of anything different.
Thanks. Next, we have a question in spinal cord injury field. If you have a transected cortical axons, can stem cell therapies improve the regeneration of these axons?
Yeah, there's been a lot of there have been a lot of efforts that have gone towards this very idea, starting from back in the day. I think even more than 2 decades ago. What I would say is this: the evidence right now suggests that if you take some of these grafts, so maybe not even necessarily just from stem cells, but also just neural stem cells, they can stimulate some kind of growth from these transected axons. So these damaged axons will grow into the graft. They do seem to make connections with these immature neurons that have been grafted there. However, they don't grow beyond the graft yet. At least there haven't been any, I don't think there's any evidence for that they can grow into a graft and then through the graft to the other side of the lesion. And I think that might just be because all of these kinds of treatments are going to be multifactorial. So simply just the graft alone might not make it. You might have to sort of stimulate the axons themselves. Another idea could be that perhaps we've given them the wrong kind of grafts. So maybe they're forming synapses or connections with cells, and that's the reason why they're not growing. So had we given them other cell types, they might have been able to grow through. The evidence so far at least suggests that you can stimulate some kind of growth from these damaged cortical axons, but it's not long distance. And to what extent does that contribute to functional recovery is anybody's guess.
Thank you. And I know we're running over, but I do have a couple more questions just to get to, if that's okay with you.
Yeah, sure.
Great. So one question about what is the effect of age on stem cell potency?
Yeah, that's a great question. There's definitely, so this is actually work that was going on at Harvard while I was there. You can actually do something called parabiosis. And this is work that was done by Amy Wagers and, oh God, I'm blanking on his name, Lee Rubin. And you can look that up. So this is really cool. You can take a young mouse and an old mouse, and you can sort of join their circulatory system so that now the old mouse is getting the young mouse's blood and vice versa. And it turns out that with age, for instance, our adult tissues lose the number of stem cells that reside there. You lose the number of stem cells, you also lose how many of them actually maintain their stem cell nest. So because over time, you lose some of these cells either through death or they differentiate and they're no longer stem-like. And the cool thing about this was that you could take young blood, infuse it into the older mouse. Yeah, it almost sounds like a Dracula movie, but you can make the aged adult stem cells and start to behave as if they were in a younger animal. And they've found that there are actually extrinsic factors, something called GDF11, that can revert some of this. Now, the evidence for this in the nervous system is a little bit limited, but I think Lee Rubin's lab had some data that showed that at least in the hippocampus, which is a part of the brain where there are stem cells normally that are giving rise to new neurons, that this holds true, that you can actually maintain stemness for longer by taking these extrinsic factors that are circulating in the young mouse versus the old. But that's a great question. There's definitely the controversy that I don't want to go into. Basically, to what extent does this happen in humans? But I think in the interest of time, I'll just save that
for the next webinar.
Yeah.
Question about, just to try to get a little bit of an understanding. So at what rate do this stem cells, and I'm probably going to I'm going to assume they're talking about embryonic stem cells, divide and multiply, say, in comparison to, say, like a cancer cell?
Yeah. So that's actually a really good question. Part of it also depends at what stage the stem cell is. So embryonic stem cells do tend to divide more quickly. So even if you think about this at the level of the embryo, right? Those initial divisions have to happen more quickly because you have to develop more numbers of cells. And then, for instance, when you get to specialized tissues, within, for instance, that cartoon that I showed you through development, you first have more copies of neural stem cells and then the numbers sort of decline. Through development, there is a decrease in that kinetics. So the cell division slows down. The idea is that potentially some of this is because now you're getting to more specialized kinds of differentiation. So you don't want the cell to undergo division too quickly, because then some of these molecules that have to be partitioned in specific compartments, that won't happen. So the short answer to your question is, early on, embryonic stem cells would probably be dividing at the rate at which cancer stem cells do, or cancer cells do. But later on, this division rate goes down. But the interesting thing about this also, and the complexity keeps going on. Even in cancers, not all cells divide at the same rate. And so this has led to the idea that there are actually things called cancer stem cells or brain tumor stem cells, where you have these stem cell populations that actually don't divide as quickly. They give rise to other cells that divide more rapidly and expand the tumor. So when you have irradiation, etc., you ablate all the rapidly dividing cells, but the true cancer stem cell is somehow resistant to this kind of therapy. And so then when you get recurrence, those are the cells that aren't actually dividing very quickly that actually give rise to recurrence of cancer.
Excellent.
That was a long question. Yeah.
No, it's good. And we're going to try to get to at least 2 more questions. So, neuronal function often requires interactions, as you pointed out, between the neurons and the glia cells. And has there been anything studied, or maybe you want to just revisit that kind of talks a little bit about that, or what the latest findings are?
Sure. So actually, another interesting, great question. So it turns out that to maintain, especially in the realm of adult stem cells that sit inside the brain, they have to reside in very, very specific niches. And so it turns out that to maintain a stem cell, it takes a family. So a stem cell does not maintain itself as a stem cell in isolation. There have to be specific factors that it has to see to maintain its stemness. So it maintains there's a very tight niche. And this not only includes glial cells, but it actually also has some of the endothelial cells, so the lining of the blood vessels, for instance, that maintain a specific environment that nurture the stemness of those stem cells. So it's not just about the neuron to glial kind of interaction, but these glial cells themselves are highly specialized that seem to reside around the stem cells. And you can actually go and look at some of the work from Sally Temple up at Albany. And she has done some beautiful work looking at what exactly does that niche comprise of. So there's Sally Temple, there's Fiona Doish, who've done some work along those lines. And if interested, I can send you some of those references.
Wonderful. And I think we're really over time. So what we can do is we can get you some of these questions, and we can touch base with the people who have sent them in, so we can get those questions answered. But I just wanted to thank everybody. We at Burke Neurological Institute would like to thank everyone for their time and their participation today. We will email the recorded webinar to all the